Good morning and welcome back. So, what we're going to do in this final vignette that comes from our series on scaling is first we're going to examine a couple of key lessons that are going to transcend this particular example. And then I'm going to try and give you a hypothesis. It's really just that. It's a plausibility argument for the necessity for mitochondria in larger cells. So, before doing that, I had a couple of, let's call them lessons. And the first one has to do with the nature of competition. And my logic is that lots of stuff that we see out in the world is the result of competition between factors which tend to drive the system in different directions. So let me give you an example. An example would be if I have some system and in it there's a receptor, and then I also have some ligands. And the properties of the binding, so I could draw a curve, which is concentration of receptor, and this is P bound, probably that the receptor is occupied, and it will undergo a curve kind of this variety. So this is known as a Langmuir binding isotherm, and I can even write the equation. So P bound as a function of concentration of ligand can be written simply in the form C over KD over 1 plus C over KD. And you can see that in the small concentration limit, this thing has a slope it's linear and has a slope of 1 over KD. And in the large C limit, it saturates to a value of 1. What does this have to do with competition? Well, the binding of these ligands to the receptor is a competition between entropy on the one hand, which tends to want to leave the ligands in solution, and binding energy on the other, which tends to want them to undergo the sort of Velcro-like reaction between a molecule of interest and the receptor. So that's one example. Another example, so I guess I'll call this example 1, and this is writ large, it's entropy competition. The second example will be surface to volume. And an example of that might be that a second phase might precipitate in a solution. And in order to do so, it may be true that the bulk free energy of let's consider a spherical precipitate, the spherical region will have a lower bulk free energy, but there's a cost to be paid, which is an interfacial free energy. And so there's a competition between, on the one hand, the gain in free energy due to the bulk term, and then the reduction in the free energy, or I could, yeah, a sort of a penalty for having the interface. So that's the first thing that I want to say. Writ large, these surface to volume competitions often will require us to remember that the volume scales with the radius as r cubed, and the area scales with the radius as r squared. And what that means is that as the volume gets larger and larger, the volume to area ratio it has units, but it's gonna grow with increasing r. So that's the backdrop to what I want to talk about. So in light of that, now what I want to think about is I want to think about the following simple question, shown here, which is, let's imagine that cellular cytoplasm costs a certain amount of energy. And I highlight that energy here, which I'm expressing as, on the left, is 10 to the 6th ATPs per micron cubed per second. So the argument being made is that there's a certain cost to run the cellular economy, just like there's a certain financial cost to each of us running our monthly budget in terms of dollars or whatever our units might be. And so there's a bulk cost of 10 to the 6th ATPs per micron cube per second. Then in addition, what's happening at the surface is that every ATP synthase is, for the purposes of our argument, going to be thought of as synthesizing on the order of 100 ATPs every second. And the question is, as I make the cell larger and larger, that means there's more microns cubed that needs to be fed with ATP. And our question is, how big does the cell get before the surface to volume ratio problem becomes so acute that the ATP synthases cannot keep up with the demand for ATP in the bulk interior? And this same kind of thing is going to show up later when we think about compartmentalization of enzymatic reactions. In general, this notion of the surface to volume ratio and the associated competition is a very deep and interesting one to consider. So, with that in mind, what I want to do is I want to try to justify what we see here, which is from Mike Lynch and Marinov in their PNAS paper on the biosynthetic cost of a gene, or some title like that. And what I want you to notice is on the y-axis, this is in units of 10 to the 9th molecules of ATP per cell. And so, as we go up, we get more and more ATPs. On the x-axis, we've got the cell volume. And so you'll recall that the volume of E. coli is around 1 micron cubed. And that means that the cost is, for this is maintenance cost per hour. So, it's something like, let's just call it 10 to the 9th ATPs per hour. But then I can divide that by few hundred seconds per hour, and let's do that. So I'm going to make the claim that we have 10 to the 9th ATPs per micron cubed per hour times 1 hour is few times 10 to the 3 seconds. That leaves me with 1 over few times 10 to the 6th ATPs per micron cubed per second. And that in turn is going to lead us to something like few times 10 to the 5th ATPs per micron cubed per second. Remembering that 1 over few is the same as few over 10. That's the rule of thumb that I used. Okay, so that's the claim. The claim is that the cost to run the cellular economy for maintenance is going to be something on the order of, I'm going to call it a 10 to the 6th ATPs per micron cubed per second. Now let's see if we can understand where such a claim comes from. Let's recall that the number of proteins per bacterium is on the order of few times 10 to the 6th. So, what we're going to claim is that here, few times 10 to the 6th proteins per micron cubed bacterium. So that's how many proteins there are. You can see for faster growing cells, it's actually a factor of 2 above that. So the cost of protein synthesis alone, we can estimate how, by remembering that we have 3 times 10 to the 6th proteins per cell, or maybe I'll write it per micron cubed, times 3 times 10 to the 2 amino acids per protein, times 4 ATPs per amino acid polypeptide bond, and this is all divided by 1,000 seconds. This is in the case of fast division, which leaves me with something like, so few times few is 10, so this is 4 times 10 to the 9th divided by 10 to the 3, so 4 times 10 to the 6th ATPs per micron cubed per second. So that's my rationalization. So my rationalization for what we saw in the Lynch and Marinov thing. So, here we were looking at this region. We see that, there's a certain cost in ATPs per cell, and depending on the cell volume, here we fixated on this value, which was kind of typically bacterial, and now we've justified the idea that that costs around 10 to the 6th ATPs per micron cubed per second. This is for the case of dividing cell. So that would be more up here for growth. So a factor of 10 down from that gets us back to that few times 10 to the 5th. something like that. Okay, so now what we want to do is I want to undertake this question. The question is very simply expressed. How big would a cell need to be, a spherical cell? We're going to consider a spherical cell. How big would it need to be before the surface was no longer able to deliver the needs of the bulk? And that balance is going to occur when the bulk, so the limit will be when bulk consumption is equal to the surface production. That's the critical condition, and any radius larger than that, the interior of the cell will not be properly stoked with ATPs and thereby able to keep up. So bulk consumption, I'm going to say R bulk times V, and that's going to be equal to sigma, which is number of ATPs per unit area, and then times R synthesis or synthases, how many ATPs are made per synthase, and then times the area. So this tells me R bulk times 4/3 pi R cubed is going to be equal to sigma R synthase times 4 pi R squared, and that leaves me, and I'm interested in trying to figure out the radius. So these two conditions together, or this condition, is the same as saying R is equal to 3 sigma R synthase divided by R bulk. So let's be clear again as to what we've done here. We've said we're going to seek that condition under which there's maximal surface production of ATP, and then we're going to match that to the rate at which ATPs are consumed in the bulk. There's a certain radius at which that condition, the maximal surface production, is equal to the rate of bulk consumption. And this condition that I wrote here is that condition. Now we need to think about things a little bit more. So what was this sigma? Sigma was defined as the areal protein density. And I'm going to show you some fun data on that. And here it is, right here, this graph. So this comes from mass spectrometry, and what I want you to notice is the y-axis tells us the inner membrane protein density in units of femtograms per micron squared, and then this axis is growth rate, but what we see is that the areal density of proteins is basically constant, and I'm going to call that few femtograms per micron squared. So let's think about that for a moment. So areal density of proteins, membrane proteins. Let's think that through. Areal density of membrane proteins Well, I just said it's 3 femtograms per micron squared, which is equal to 3 times 10 to the minus 15 grams per micron squared. If I want to find out how many proteins that is, then I'm going to take 3 times 10 to the minus 15 grams per micron squared and divide by 3 times 10 to the 4 grams per mole. Why? Because 30,000 Daltons is like my typical number for membrane for proteins in general. This is probably an underestimate for membrane proteins, but it doesn't matter. A factor of 2 here and there is not relevant to my basic endeavor here. So this leaves us with, let's see, this leaves us with, I'm going to call it 10 to the minus 19 moles times 6 times 10 to the 23 molecules per mole, which leaves us with 6 times 10 to the 4th per micron squared. You see what we've done? We've basically estimated the number of proteins per unit area And indeed, the rule of thumb that I carry around in my head is 100,000 per micron squared. That's a very typical membrane protein number. So we now know this sigma. So I'm going to call this 10 to the fourth proteins, this is membrane proteins, per micron squared. So that's the areal density. So now we can work out this R. So R is going to be, we're estimated as 3, and then times 10 to the fourth proteins per micron squared, and then R synthase is 100 ATPs per protein. Maybe I should have used a different notation. Let me call this 100 ATPs per synthase per second. And instead of writing proteins over here, let's call that synthases. So I'm maximally packing synthases, and then I divide that by the bulk rate of 10 to the sixth ATPs per micron cubed per second. Look at the units of this thing. So let's see. Just kind of curious about one thing. Oh yeah, I think I made a mistake, didn't I? The sigma is 10 to the fifth, not 10 to the fourth. I apologize. So I just got sloppy. So now this makes a lot more sense. So so what we find is that this value is something like 30 microns. Because I have 10 to the fifth times 10 to the 2, that's 10 to the seventh divided by 10 to the sixth, leaves me with 3 times 10. So some number, usually when I do this estimate, let's see, I actually did it a little while ago this morning, so what was the difference? Yeah, the difference was this 4, the 4 times 10 to the sixth ATPs per micron cubed per second for growth. But that's fine. I'm not gonna worry about this. So let me write in red. You could have put a 4 here, and then this would have been divided by a 4. So either way, it's just an order of magnitude estimate to try to make a point. So what do I learn from this? So my claim is that this figure, this toy figure, is a conceptual model that we have of a simple competition between surface and volume effects. And the competition plays out because the surface is producing ATPs, which are in turn being consumed within the bulk. We find that once the radius is between 10 and 30 microns, the surface decoration with ATPs is no longer able to keep up with the demands of the bulk, and that leads to the hypothesis, if you will, for the existence of mitochondria, because you might remember that mitochondria are these little factories that have very, super interesting twisted membranes, and in those systems, they're packed full of ATP synthases, but instead of having to exploit the whole plasma membrane area, the area is confined to these organelles. And in fact, a very interesting exercise for all of you to think about is how much area is available inside of all the mitochondria, and how does that compare to the plasma membrane. So I think this is really all that I had to say about this particular exercise. It's again just to try to show you a style of thinking about scaling estimates, and here